Hello everyone, my name is Ellen. I'm from São Paulo, Brazil. I hold a master's degree in pharmacology from the University of São Paulo and a Ph.D. in toxinology from Butantan Institute. I have been working with rattlesnake venom for the past decade, so I'm so very thankful for this invitation. Thank you, Sophia, and I'm grateful for this opportunity to share my knowledge of these toxins and my experience. So today, my masterclass is about rattlesnake venom from toxicological effect to drug discovery. So I want to change your perspective of how something that is toxic and lethal could be a potential therapeutic drug. We'll start this masterclass by questioning why do snakes produce venoms? So snakes have venom as an evolutionary adaptation for hunting and defense. The snake venoms are complex mixture of biologically active compounds, mainly comprising proteins and peptides secreted from specialized venom glands. So snakes have developed this ability over millions of years as a evolution, so they can enhance their ability of survival and hunting success. So the venom provides the snakes a significant hunting advantage. So when snakes inject their venom, it contains these mixtures of these specialized toxins that can help them to immobilize it and kill prey so they can have efficient hunting and digestion. So snake venoms also serve as a defense mechanism. So when a snake feels threatened, it may bite and release the venom as a mechanism of defense. So what the snake venoms are composed of? So in these pictures summarizes the main snake venom components. Most of them are composed by proteins and peptides, which can be 90 to 95%. And it can be classified as non-enzyme and enzyme, so depends by their intrinsic enzymatic activity. The 5% left is the non-proteic component, which can be lipids, amino acids, ions, carbohydrates, nucleosides. So by the proteomic analysis, have demonstrated that snake venom contain proteins from a wide range of family, about 26 different proteins families. So this composition of different proteins families is significant for the variation of the snake venom in different snake species. So depending on the composition of the venom is unique for a specific snake species. So this is important to understand the venom components, because it's an important tool to study the physiological process in humans, and as well for the clinical diagnosis. So understand how these proteins works on the body, which can help for the clinical diagnosis. So since snake venom present high complexity and diversity, this results in a variable biochemical and toxicological profile, and this determines a wide range of clinical manifestation. So some toxins can cause tissue damage, local manifestation, for example, necrosis. So these lead a permanent consequence. And some other toxins can cause systemic effects, for example, neurotoxicity, bleeding, acute kidney injury, rhabdomyolysis, cardiotoxicity, and thrombosis. This diverse array of toxins contributes for these many clinical manifestations presented in snake bite envenoming, and this highlight how complex is snake venoms and its severity during the snake biting envenomation cases. And other thing is that snake venoms, since they present these biologically active compounds, this is important because they serve as a tool for pharmacological application, because they present the main characteristic of a drug, which is the high affinity and high selectivity towards their targets, which is crucial for influence the main aspects of the physiological process. So, according to the Ministry of Health of Brazil, the venomous snake genera with public health relevance are Bothropus, Crotalus, Mycurus, and Lachesis. Bothropus genus is responsible for about 70 to 75% of snake bite accidents in Brazil, making it the most prevalent cause of the envenomation. Crotalus genus follows as the second most common of the snake bite envenoming, and the Mycurus and the Lachesis genera contribute to only a small number of the cases. Although the Bothropus accidents are the most frequent, Crotalus accidents have a higher lethality index, and this is because of their composition of the venom, as I mentioned it before, that is species specific, and I will show you why the Crotalus have this high lethality compared to Bothropus. I will explain later. So for Crotalus genus, it has been identified about 70 species and subspecies. Here in Brazil, the rattlesnake species Crotalus dorissus is divided into 7 subspecies, which we can find in every Brazilian territory. But there are two subspecies very well known by their composition and the clinical manifestation, which is Crotalus dorissus terrificus and Crotalus dorissus colineatus. Now we are going to talk about crotalic envenoming. So during crotalic envenoming, it is classified according to the clinical manifestation. So it can be classified as mild, moderate, and severe cases. In mild cases, there is a discrete neurotoxicity signs with or without myalgia, which is muscle pain, and no alteration in the urine color. In the moderate cases, it is characterized by the present or discrete neurotoxic signs, discrete myalgia, and discrete myoglobinuria. And in the severe cases, the neurotoxic signs and symptoms are more intense and evident. Myalgia is also intense and widespread, and the patient presents dark urine, oliguria, and anuria. So recognizing the signs and symptoms of the crotalic envenoming is crucial for determining the appropriate treatment, which involves administration species-specific antivenom. So as soon as possible, as soon as these symptoms and signs are recognized, the early intervention plays a crucial role to make sure that the neutralization of the venom was very effective and saving the patient's life. So what are the signs and symptoms of rattlesnake envenoming? In terms of local manifestation, some patients typically experience mild or absent pain, along with the possibility of erythema and paresthesia. So the lack of local inflammation, local symptoms, is one of the main characteristics of clinical diagnosis of crotalic envenoming, which can be distinguished from other snake bites. Rattlesnake venom is known by its neurotoxic effect, which is myasthenic facies. Some people call drunk face because the patient could not open his eyes properly, which is called palpebroptosis. Also, the patient presents flaccidity in the muscle of the face. The patient presents pupillary diameter alteration, could present blurred vision and diplopia. So this neurotoxic effect is caused by the blockage of the neuromuscular junction. So this leads to progressive muscle paralysis and could lead to asphyxia and consequently death. The crotalic venom also presents myotoxic activity. So it causes the damage of the skeletal muscle and promotes the release of some enzymes and myoglobin. These enzymes and myoglobin are released into the bloodstream, and this leads to the presence of dark urine, known as myoglobinuria. In more severe cases, can cause acute kidney injury, and also in the severe cases can cause intense and generalized muscular pain, which is called myalgia. So now we are going to find out what are the components of the crotalic venom that was responsible for the clinical manifestation that I mentioned before. So the crotalic venom consists in 4 toxins, which is crotoxin, crotamine, gyroxin, and convuxin. Crotoxin is the most abundant toxin present of the crotalic venom. It corresponds to 60% of total venom, and it's a potent neurotoxin which inhibits acetylcholine release at the neuromuscular junction. Also, crotoxin presents cardiotoxicity, nephrotoxicity, and myotoxicity effects. Crotamine is a non-enzymatic polypeptide which exerts myotoxic and neurotoxic action by inducing cell death in skeletal muscles. Gyroxin consists in serine proteases and constitute about 2% of the total venom. Gyroxin demonstrates to be thrombin-like, to have thrombin-like properties, decreased blood pressure, and present procoagulating effects, and as well hemotoxicity. Convuxin is a glycoprotein from type C lectin. It's a non-enzymatic molecule and has a strong platelet activation and aggregation activity. So as I mentioned before, crotoxin is the major component of the crotalic venom, and it was the first toxin to be purified and crystallized. later studies have found that crotoxin is actually a complex composed by two subunits. The one subunit is called phospholipase A2 or crotoxin B, and phospholipase A2 is responsible for the toxic activity of the crotoxin. And the other subunit is called crotapotin, with no enzymatic and no toxic activity, but crotapotin potentiates the phospholipase A2 toxicity because it works as a chaperone protein. So several isoforms of both subunits have been isolated and characterized, showing that crotoxin is in fact a random mixture of a combination of these subunits isoforms, so leading to different pharmacological and biological properties that has been reported for crotoxin. Following the initial crystallization of crotoxin, it took about 13 years for researchers starting conducting pharmacological studies for this toxin. The studies provided insights about the mechanisms of action of crotoxin, as well the potential for clinical application for this toxin. So these studies highlighted the importance and to understand how crotoxin works during venomation and how crotoxin could be a potential for drug developing. Besides the neurotoxicity and cytotoxic effects, crotoxin also present a range of actions. So during these last years, crotoxin has an anti-tumoral effect, antibacterial, anti-inflammatory, immunomodulatory action, as well antiviral, and present as an analgesic effect. So crotoxin, as I mentioned before, present toxicological effects, which includes neurotoxic, myotoxic, and cardiotoxic effects. So crotoxin inhibits acetylcholine release at presynaptic level of the neuromuscular junction, and resulting in muscle paralysis and death by asphyxia. The myotoxicity of crotoxin can cause systemic and irreversible skeletal muscle damage. This systemic myotoxicity is accompanied by increased myoglobinuria and could lead to acute renal failure. For cardiotoxic effect, crotoxin decreased the contractile force of the heart and increased the creatine kinase activity. Now I will start talking about the pharmacological effect of crotoxin that has been identified over these years. The first, I will start with analgesic effect. So crotoxin inhibits both acute and chronic pain, and this effect is mediated by muscarinic, alpha-adrenergic receptors, as well as serotoninergic and noradrenergic systems. Crotalfin is a 14 amino acid peptide that was purified and characterized from the Crotalus durissus terrificus venom. It's important to mention that crotalfin, the amino acid sequence of the crotalfin is identical to the gamma chain of the crotapotin. So crotalfin is capable of inducing a potent and durable antinociceptive effect in acute pain and chronic neuropathic pain by activation of peripheral opioid receptors. Clinically, crotoxin has been tested in patients with advanced cancer, and the the analgesic effect of crotoxin was observed in 78% of the patient with solid tumor. Other studies have found that crotoxin presents antibacterial effect in both gram-positive and gram-negative bacteria, and the effect is correlated with the enzymatic activity of phospholipase A2. But more research is needed to better understand the main mechanisms involved and the potential application for the antibacterial activity. Crotoxin also presents antiviral effect in dengue virus and the yellow fever virus. It's believed that crotoxin disrupts the virus lipid bilayer envelope and impairing with the virus replication and spread. Regarding hepatitis C virus, crotoxin inhibits virus entry and release, but there is no effect on virus replication. So in the last 30 years, several studies have demonstrated that crotoxin modulates immune and anti-inflammatory response. So these studies have demonstrated that crotoxin inhibits both vascular and cell components by reducing the number of circulating leukocytes, and this was correlated with the increased releasing of cytokines, interleukin 6 and 10, which is responsible for mediating and regulating inflammation. Also, crotoxin suppress the secretion of pro-inflammatory cytokines, and recently, 10 years ago, it was demonstrated that crotoxin presented long-lasting anti-inflammatory effect when compared with classical anti-inflammatory drugs. So crotoxin modulates the macrophages, neutrophils, and lymphocytes function. For macrophages, crotoxin presents a dual effect by inhibiting spreading and phagocytosis and stimulating the stimulation of respiratory burst, which is the release of reactive oxygen species, which is toxic for some pathogens, and this changes is accompanied by the modulation of the macrophage metabolism, so accompanied by higher consumption of glucose and glutamine. So crotoxin for neutrophils inhibits both migration and phagocytosis by inflammatory agents, and this inhibitory effect of crotoxin on neutrophil functions contribute for its anti-inflammatory effect. Additionally, crotoxin modulates the neutrophil-macrophage interaction, and regarding lymphocytes, crotoxin decreased the peripheral trafficking, decrease lymphocyte proliferation, and as well antibody production. Later finds have demonstrated that this anti-inflammatory and immunomodulatory effect of crotoxin was mediated by the release of lipid mediators such as lipoxin and its stable analog, which is associated with pro-resolving, which have pro-resolving properties, and there is also a participation of the formylpeptide receptors. This mechanism of releasing lipid mediators and the involvement of formylpeptide receptors by secreted macrophage was correlated with antitumoral and anti-angiogenic action of crotoxin, because since the secretory activity of macrophage was modulated by crotoxin, this resulted in inhibition of tumor cell proliferation in vitro studies. But in vivo, it was observed as well a reduction of the tumor cell growth, and this effect was associated with the decrease of the formation of new vessels, and as well this reduction of tumor mass was correlated as well with the analgesic effect of crotoxin mediated by these lipid mediators, by this mechanism. So crotoxin, when macrophage was treated with crotoxin, it demonstrates as well reprogramming macrophage phenotype from pro-angiogenic to an anti-angiogenic phenotype, and crotoxin interferes as well with the phenotypic reprogramming of the macrophages on tumor microenvironment. So, in conclusion, for this part, several studies have shown that crotoxin modulates both events of innate and humoral immunity by producing endogenous lipid mediators with anti-inflammatory and immunomodulatory, as well as anti-tumoral activity of crotoxin. So crotoxin is a possible there is a possible medical application of crotoxin against many human disease, but I don't want to say that crotoxin is a potential drug, but a potential drug design. So studying the main biological effects of crotoxin in some disease, it's possible to make this toxin as a drug design. So we are going to talk about my favorite part of this presentation, which is the anti-tumor effect of crotoxin. So this anti-tumor effect can be divided in three main aspects. So crotoxin can have a direct effect on tumor cells, so influencing their behavior, and it may inhibit the tumor growth. Also, crotoxin modulates the tumor microenvironment, so creating unfavorable microenvironment for tumor progression. And there is ongoing clinical trials studies which use crotoxin in patients with advanced cancer. So this study is in phase 1 and have demonstrated promising results. So crotoxin in this patient was well tolerated by their neurotoxic effect is well tolerated in this patient. Also, these studies have demonstrated the potential efficacy of crotoxin in reducing tumor mass. The study demonstrated a reduction of 50% in some patients. So these findings in clinical trial highlighted the potential of crotoxin as a therapeutic candidate for cancer treatment. So as I mentioned before, crotoxin demonstrated induction of apoptosis, induction of cell cycle arrest, and autophagy, as well the decreased migration in different tumor cells types. The one possible mechanism involved is the epidermal growth factor receptors signaling pathway. So crotoxin, it was demonstrated to inhibit the ERK phosphorylation, which is a crucial protein from MAP kinase signaling pathway, and this protein regulates all these processes that it has been observed the effect of the crotoxin. So inhibition of ERK phosphorylation suggests that crotoxin is a great promise as a potential target for anti-tumor therapy. So when we discussing the tumor microenvironment, it's important to consider not only tumor cells, but also the resident infiltrating cells, such as fibroblasts, immune cells, and endothelial cells, as well the secreted factors and the extracellular matrix. So the tumor microenvironment plays a crucial role for tumor growth and progression, since the tumor can modify this environment, and in turn, the microenvironment can impact on tumor behavior, so inducing the tumor growth and metastasis. One important process is angiogenesis, which is the formation of new blood vessels. So endothelial cells in this microenvironment provide nutrients and oxygen to support tumor growth. On the other hand, there is epithelial mesenchymal transition, which is a process when tumor cells lose their epithelial characteristic and acquire migratory and invasive phenotype. So this transition is influenced by cancer-associated fibroblasts, which are the main component of the tumor stroma. So understanding the interactions between tumor and tumor stroma, it's important to develop effective therapeutic strategies. So that's why I study in this process, these two processes in my master and in my PhD research. During my master research, so we have investigated the effect of crotoxin on endothelial cells and how the toxin can impair with tumor angiogenesis. So our finds have demonstrated that crotoxin inhibits the endothelial cell proliferation, cell adhesion, and migration, especially in tumor conditionated medium. So in this condition, demonstrated even more drastic effect on the inhibition. So crotoxin also impaired the tube formation in matrigel. So in a 3D condition, crotoxin inhibited the tube formation, and this mechanism is mediated by integrin signaling pathway. So we investigate which proteins of this signaling pathway are impaired by the action of crotoxin. So these main molecules is responsible for the adhesion, the migration, formation of the lamellipodiums, phylopodiums, all the structures of actin that promote the cell migration in how can I say, in a matrix, for example, collagen or fibronectin. So our suggestion is that crotoxin impairs with the integrin signaling pathway, not by binding the receptor, but interfering with some phosphorylation of these downstream proteins. So that's why we found this, we observed this effect of the decrease of proliferation, migration, tube formation, and all these contribute for the anti-tumor effect of crotoxin. In my PhD project, we want to investigate the modulatory action of crotoxin on tumor stroma interaction. So we want to see if crotoxin could modulate the fibroblast activity and see if the EMT process was also affected by crotoxin. So one of the main markers of the fibroblast differentiation is the expression of alpha smooth muscle actin. So crotoxin in our results demonstrated inhibition of the expression of alpha SMA, indicating that crotoxin inhibits the fibroblast differentiation. Later, we investigated that crotoxin inhibits the key markers of EMT proteins, such as E and N-cadherin, alpha V integrin, and the alpha SMA, as I mentioned. And also crotoxin inhibits the spheroid invasion into collagen gel. So we used this model of spheroids composed by tumor cells and fibroblasts, and we observed that crotoxin inhibits the invasion into the collagen matrix, simulating in vivo condition. And this mechanism was accompanied by the decrease of metalloproteases 9 and 13, which degrades the collagen matrix. So crotoxin inhibits also the degradation of this matrix, and also observed that some of cytokines involved with tumor interaction, angiogenesis regulation, extracellular matrix interaction, and immune cells recruitment. So crotoxin in this project demonstrates a potential modulator of the signaling cascade involved in the progression of epithelial-mesenchymal transition, which is a critical step in the metastasis. For this presentation, I tried to highlight the potential therapeutic application of rattlesnake toxins, especially crotoxin, because since it's the major toxin found in the Crotalus duris osteificus venom. But other toxins, as I mentioned before, such as crotofin, crotamine, and diroxin, also present therapeutic application, many therapeutic application. And for crotoxin, much studies are necessary, for example, purification, isolation, and identification of specific parts of crotoxin molecule. This also includes the combination of the crotoxin subunit, since there is 4 isoforms of each subunit. So it's important to investigate which are the best match of the isoform to make the complex crotoxin more effective, clinically effective. So there is much more work to do, so with all this information, these studies of purification, isolation, and identification, we can modify crotoxin molecule to make it more effective molecule with less side effects, and maximizing the therapeutic potential of the crotoxin, and make it improve the safety and efficacy in clinical studies. So I want to thank you for watching this masterclass. These are some references I used for this presentation, and these are the references I used from our lab group. which studied crotoxin since the 90s. So I invite you to check all this work published. And once again, I want to thank you for watching this masterclass and see you soon.